Off here last time talking about the vocab word trait. Trait is about length of small toes, or the toe next to the big toe. We talked about hairline, and so a trait is a visible characteristic of an organism. A visible characteristic of an organism. Now, there's several other vocab words that we need to understand before we can do genetic crosses. Purebred is another vocab word that you'll need to know. And purebred also is called true breeding. And a purebred organism is one in which all the offspring produce the same trait over many generations. And we could have said here that a purebred creature is homozygous, either homozygous dominant or homozygous recessive. In this case, all the creatures, all these petunias or pea plants, are homozygous dominant. They're big A, big A. And they're crossed with another plant that's big A, big A. They are purebred. They will only produce offspring that are homozygous dominant. Okay? So that's purebred. Phenotype. Phenotype is a word that I've used already. And phenotype refers to the expressed trait in an organism, or what it looks like. It doesn't refer to the genes. It refers to what the genes code for. In this case, blue eyes versus brown eyes, or wrinkled seeds versus round seeds. Okay? So yesterday we talked about the recessive homozygous recessive genotype, little b that codes for blue eyes. And that's recessive to the dominant allele that codes for brown eyes, big B. Genotype, on the other hand, refers to the whole genetic composition of an organism. And it is the ACTA letter combinations for all the genes that are used to represent it. And it makes up the chromosomes. So genotype refers to the chromosomes and the genes that are found on those chromosomes. It refers to the base pairs, the A's, the T's, the C's, and the G's. Okay? All right. Gene, on the other hand, is a segment of DNA that codes for one and only one protein. I'd like you to write this definition down. Gene, a segment of DNA that codes for one and only one protein. A gene is several hundred to several thousand base pairs long. A base pair, remember, is an A and a T or a C and a G. That's going to be important for you to understand the definition of a gene. Okay, allele. I'd like you to write this down too. Allele. There are several alternative forms for every gene. 2 or more copies for every trait, in fact. And allele corresponds to the double-stranded nature of DNA. So one gene, one series of several hundred to several thousand base pairs, has 2 alternative forms. One may be dominant, one may be recessive. So every trait has 2 alternative forms called alleles. They both may be dominant, they both may be recessive, or one may be dominant and one may be recessive. Okay? It all depends on what they have inherited. They might have two different alleles, or they might have the same allele, whether it's dominant or recessive, big A, or big B, big B. And those alleles are on the two chromosome pairs. So in this slide, the upper chromosome has an allele for phenotype A, might be coding for blue eyes, little b, little b. The lower chromosome has the allele coding for phenotype B. It might be brown eyes, it might be big B, big B. Okay. Does that make sense, guys? Okay. Dominant and recessive. The allele that is expressed in the phenotype and masks the recessive allele is called dominant. If you like, you can copy this one. We might go kind of fast at this point. So dominant allele is the one that is expressed in the phenotype and it masks the recessive allele. If this one's present and there is a recessive allele, the recessive allele will not show up in the phenotype. So the recessive allele is the one that does not show up in the phenotype because it is masked by the dominant allele. So I gave you a couple of examples on this slide. The one on the lower left is with flower color. In the flower color example, there is a purple homozygous dominant flower crossed with a white homozygous recessive flower. The purple is dominant over the white. So one flower contributes a purple allele, a big R. The other flower contributes a white allele, little r. When they're together in the offspring, the purple flower dominates, and all the offspring are purple, even though they have the allele for white flowers. In the offspring there, it's big R, little r. Even though they have the gene or the allele for white flowers, the flower is not white. It's purple because purple is dominant. In the pea pod example, both pea pods are heterozygous. Green is dominant over yellow. So two green heterozygous pea pods are crossed together, and in the offspring, 3/4 of them are green, and 1/4, the homozygous recessive, is yellow. When the big G is there that codes for green, even though the gene, little g, that codes for yellow is there, it's green. The only time that the yellow shows up is when it is two small g's, two alleles that code for yellow, with no dominant big G allele there. Haploid. Don't write this down. Haploid. A cell that has gone through meiosis, that's meiosis, not mitosis, and having half the normal amount of chromosomes. So, for example, sperm and egg. For humans, it's 23 chromosomes. The haploid condition for humans is 23. Okay? Diploid. Don't write this down. Diploid. A cell that is going through meiosis and has doubled the normal amount of chromosomes. Doubled the normal amount of chromosomes. So for humans, I'll show you a picture of what that looks like. Okay, here's a picture of meiosis or reduction division and haploid and diploid. Here is the haploid condition, 2N. There's 2 copies of every chromosome. Those copies duplicate and create a diploid situation. Okay, diploid. Once those diploid cells divide, they create haploid sperm in this case, and if it were over here, it would be haploid eggs. Okay? So here it is in numbers. It's sometimes called 2N and 4N. Here it is in pictures showing what it looks like. Chromosomes replicate. They form double the normal amount, or 4N. Then it divides to 2N, and then it divides further to form haploid cells, sperm and egg in this case. Okay, does that make sense? Okay, let's watch a video on how this works, and we'll review some of the terms. So during a video, if you see
Our possibility to explore the DNA sequence of the human genome has made...
If you see stuff that you think you should write down...
Hi, my name is Mary Pockema. I'm an adjunct.
If you see things you should write down during the course of the movie, you can write it down. So yeah, you could take notes on this if you want.
Professor's biology. And today, we're going to have a little fun. My name is Mary Pockema. I'm an adjunct professor of biology. And today, we're going to have a little fun with Mendelian Punnett squares. Now, before we get started and jumping into our Punnett square problems, let's set down some ground rules. First, we're going to talk about alleles. Alleles are going to be like different flavors of genes. So, go into the ice cream shop, there's 31 flavors of ice cream. Even though they're all different flavors, they're all still ice cream, right? Alleles are going to be those different flavors of genes that you have available to you. The next thing I want to talk about is dominant and recessive. In any relationship, some of those alleles are going to be recessive. Those dominant alleles, if they're present, they're going to have more of a say in what the outcome of that process is going to be. And the recessives, well, they're going to have little to no say unless they are 2 recessives that are available. Also, in each of our crosses, we're going to have 1 allele from our mother and 1 allele from our father. Now, let's jump into our crosses. Today, we're going to do a 4-square Punnett square. So, you want to start out by just drawing a square. In that square, you're going to draw a line right down the middle from the top to the bottom and a line from left to right. Now, you have 4 individual squares. Each of those squares is going to be a probability, and each of those squares is 1/4, right? Or 25%. Next, we're going to put our parents' alleles on the Punnett square. Now, it doesn't really matter if you put...
Okay, we have a few more minutes until the fire alarm. Let me tell you what to do when the alarm sounds, because you won't be able to hear me when it does. When the alarm sounds, you're going to silently, quietly, and orderly line up around the edge of the room. The first person over here is going to be Michelle and Lauren in line, and the rest of you can line up here. When I give the okay, we'll start to move out in ninja mode, complete silence, okay, in an orderly fashion, yeah. So it depends on the type. It depends on the type. If it's an earthquake, yeah. If it's a fire, no. Okay, does that make sense?
Male or female on the top. General.
Let's get started. Let's get started. Daniel? We ready?
People put male on top, female to the left. For our problem today, we're going to use eye color. The eye color alleles are going to be, for our father, it's going to be brown phenotypically expressed alleles. That means that that father has brown eyes, but his genotype, or the genes at play, his alleles are going to be big B, little b. That means that he's heterozygous, or he has two different alleles for that trait of eye color. It's a little important to note at this time that all of these letters are completely arbitrary. You can make up any letters you want. However, in any relationship, a capital letter or a big letter, like say big B, is going to be for the dominant. And a little letter, or a lowercase letter, in this case, little b, is going to be for the recessive. Okay, so we have the father. Big B, little b. He's heterozygous, meaning he has two different alleles. What about the mom? The mom has blue eyes. So phenotypically, her expression that you see on the outside is blue eyes. Her genotype, or the genes at play, are going to be little b, or homozygous recessive. Alright, it's time to do our cross. Now, we're going to take one from the top and one from the left, and we're going to bring them down into those squares. So, we have on our top, big B, little b, then bring another big B, little b down, and our right square at the top, yeah.
Not ringing the bell, so when you're just going to stay in your first grade classes until the evacuation fire drill bell rings, okay? Stay in your first grade classes, thank you.
Little b, and our bottom right square, we have little b, probability of getting an offspring with what kind of eye color. Now remember, each of those squares is going to be 25%. So, we have a 25 plus 25 or 50% chance of getting an offspring with brown eyes. And we have a 25 plus 25 eyes. And for a fun little extra problem when you're sitting around the dinner table, take the eye colors or the phenotypes of your parents. Even though you won't know the exact genotypes or the genetic makeup behind what you're seeing on the outside, do some crosses and see if you can find out the probability that you could have actually had a different eye color. Thanks for watching, and if you want to learn more about this subject, click on the link below. Or if you want to learn more biology, feel free to click on any links around it. And please, rate, comment, and subscribe to this channel. Or maybe you have ideas for more videos, send an email at request@allele.com. Thanks for learning.
Okay. All right. So, did you guys understand how to do that cross?
Yes.
Okay. The cross using the punnett square.